Run your fingertip along the inside of your wrist right now, out where the light can reach it. If the skin there holds a warm golden cast, if it slides into a tan instead of turning red and angry under a few hours of sun, you are carrying something that scientists have spent the last several years quietly moving to a completely different part of the map. And once you hear where they actually traced it, the story you probably grew up believing about it is going to sound almost quaint. For most of the last 100 years, that particular warm, sun-tolerant skin tone got filed under one single word: Mediterranean. People pictured fishing villages on the coast of Greece, vineyards in Tuscany, the beaches of southern Spain. It felt obvious. It felt like it needed no further explanation. The same way nobody questions why people near the equator tend to have darker skin than people near the poles. The Mediterranean sun, the Mediterranean coastline, the Mediterranean diet even, all got wrapped up into a tidy little package that explained the tone of a lot of people's skin without anyone ever really checking the genetics underneath it. Then a new kind of science showed up that could actually check. Over roughly the last 15 years, researchers have gotten dramatically better at pulling usable DNA out of bones that are thousands, even tens of thousands of years old. Ancient DNA, as the field is called, used to be almost impossible. Bone degrades, DNA breaks down, contamination from modern human handling ruins samples constantly. But new extraction techniques, developed largely within the last decade and a half, changed what was recoverable, and once labs started applying those techniques to skeletons from the mountains of Western Asia, the tidy Mediterranean story started to come apart at the seams. Here is where the story actually begins, and it does not begin anywhere near a beach. It begins high in the Zagros Mountains of western Iran at a Neolithic archaeological site called Ganj Dareh. Ganj Dareh was a small farming and herding settlement, one of the places where humans were first learning to raise goats and grow crops instead of only hunting and foraging for them. This was happening at almost the exact same time that a separate, related shift toward farming was unfolding further west, in the region known as the Fertile Crescent. Two agricultural revolutions, essentially happening in parallel, in two related but distinct populations. At Ganj Dareh, archaeologists recovered the remains of a woman who lived roughly 10,000 years ago. For most of human history, she would have simply been bones. In the ground, a name and a face nobody could ever recover. But modern ancient DNA techniques let researchers do something remarkable. They sequenced her genome. They read the actual genetic code she carried in life, letter by letter, out of bone that had been in the ground for 10 millennia. And when they looked specifically at the genes known to control skin pigmentation, they found something that did not fit the assumption everyone had been working from. She carried at least one copy of a gene variant called SLC24A5, a variant strongly linked in modern populations to lighter skin. If that had been the whole story, you might expect her to land somewhere close to the pale end of the human skin tone range, similar to a lot of people in modern Northern Europe. But she did not carry the second major variant that, when it shows up alongside SLC24A5, is what actually pushes skin tone all the way toward that very pale end. Without that second variant, her genetic profile settled somewhere in between, lighter than a fully dark ancestral tone, but nowhere close to the palest end of the spectrum. In plain, everyday language, her genes were sitting in almost exactly the zone we would now call olive skin. And she was living in the highlands of Iran at the very beginning of agriculture, thousands of years before anyone had ever heard of Rome or Greece or the word Mediterranean. For a long time, the working assumption in both popular science writing and a fair amount of academic writing too was that skin color runs along something like a dial, dark at one end, pale at the other, with everyone's ancestors sliding along that single scale depending mostly on how much sunlight hit the regions where they lived. It is a simple, appealing idea, and it turns out to be too simple. It flattens skin tone into one dimension when the underlying biology is actually working in several dimensions at once. Olive skin is not just a midpoint stop on that single dial. It is its own specific genetic combination, distinct from simply being halfway between light and dark. And once researchers had ancient genomes to check against instead of just guessing from modern populations, that distinction became impossible to ignore. So, what is actually happening under the skin, biologically speaking, to produce a tone like this? It helps to start with the basics, because most people never actually learn how skin color works at a cellular level, only that it does. Human skin gets its color from a pigment called melanin. Every single person on Earth produces melanin. Nobody is without it. What differs from person to person and from population to population is not whether melanin is present, but how much of it is made and which of two types dominates. There are two forms of melanin that matter here. One is called eumelanin, and it produces brown and black tones. The other is called pheomelanin, and it produces yellow and reddish tones. Think of them like two base colors on an artist's palette that never leave the canvas completely. Every person's skin tone is really a blend, a ratio between these two pigments, combined with how much total pigment gets produced and how it gets packaged and spread out inside individual skin cells. Change the ratio, change the total amount, change the packaging, and you change the visible result, even if the basic ingredients never change. And here's the part that surprises a lot of people who assume genetics works like a light switch. Skin color is not controlled by a single gene. It is what geneticists call a polygenic trait, meaning dozens of different genes each contribute a small piece to the final outcome, working together rather than any one of them acting alone. It helps to think of it less like flipping a single switch and more like following a recipe with dozens of ingredients. No single ingredient decides what the finished dish tastes like. It is the combination, the proportions, and how they interact that produces the final result. Skin color genetics works the same way. Genes with names like MC1R, TYR, OCA2, IRF4, and SLC45A2 all play supporting roles. But two genes in particular matter, more than almost any others in this specific story. SLC24A5 and SLC45A2. Specific combinations of variants at those two genes, layered on top of each other and interacting with several smaller contributors, are what actually produce the intermediate, warm, golden-leaning tone that we call olive skin. There is a popular idea floating around online, the kind of claim that gets repeated in comment sections and casual conversations without anyone ever checking it, that somewhere out there is a single clean olive skin gene. The idea usually goes something like this. Just as some people talk about a single gene for red hair or a single gene for blue eyes, there must be one specific genetic switch that turns olive skin on or off, something a lab could point to on command. It sounds tidy. It sounds like the kind of clear, simple answer people want when they ask a genetic question. And once researchers actually mapped this trait out across large ancient and modern populations, that tidy idea did not survive contact with the evidence. There is no single olive skin gene. There is no one genetic switch a lab can flip and say, this and only this produces the trait. What actually produces olive skin is a specific combination of variants stacked across several different genes, interacting with each other in ways that only fully make sense when you look at the whole combination together rather than any one piece in isolation. And just as important as the fact that it is a combination rather than a switch is the fact that this particular combination did not stay parked in one place. It moved. It traveled across enormous distances, carried inside the bodies of people who had absolutely no idea they were carrying a genetic signature anywhere, let alone one that scientists would eventually be able to trace across 10,000 years and thousands of miles. If a trait sitting on the surface of your own skin can trace back through bones that are 10,000 years old, recovered from a mountain valley, almost nobody outside a small circle of geneticists has ever heard of. It raises an obvious question. What else, quietly written into your own DNA right now, is far older and far stranger than the family story you grew up believing? The variant found in that woman at Ganj Dareh was not some isolated fluke limited to her small community. Researchers found the same variant already circulating just to the north among a hunter-gatherer population living in the mountains of what is now Georgia and Armenia, a region scientists now refer to using the shorthand Caucasus. Hunter-gatherers. That detail matters enormously because it means the variant was already present in this general highland region of Western Asia before these two related groups, the Zagros farmers and the Caucasus hunter-gatherers, had even fully split apart from one another as distinct populations. In other words, the genetic groundwork for this trait was laid down long before either group existed as a separate, identifiable population, likely tens of thousands of years before the Neolithic period we associate with the beginnings of farming. That deeper layer became much clearer thanks to a set of ancient genomes recovered directly from sites in the Caucasus Mountains themselves. One came from a location called Satsurblia Cave, dated to roughly 13,000 years old. Another came from a rock shelter called Codius Kulda, dated to roughly 9,700 years old. Both sites sit in western Georgia. When researchers sequenced these two genomes, they revealed a previously undocumented ancestral lineage, the population now known as the Caucasus hunter-gatherers, a group that had split away from hunter-gatherers further west in Europe somewhere around 45,000 years ago, and that diverged from the ancestors of the later Neolithic farmers roughly another 20,000 years after that. And crucially, both of these ancient Caucasus genomes already carried that same derived, lighter-leaning pigmentation variant, the very same, one later found in the farming woman at Ganj Dareh. That single detail pushes the known presence of this variant in the highland region back to well before the last ice age had even ended, into a period so early that agriculture was not even a distant idea yet, let alone a lived reality. The genetic material for this specific skin tone was already sitting in the DNA of people hunting and foraging their way across an ice age landscape. tens of thousands of years before anyone in that region ever planted a single seed. This same Caucasus hunter-gatherer ancestry went on to become one of the two major genetic building blocks of a later population called the Yamnaya, a Bronze Age culture from the Pontic steppe that is closely tied to the later spread of Indo-European languages across huge stretches of Europe and Asia. Researchers involved in this line of work have also noted that the same Caucasus-related genetic component appears to be one of the closest matches found so far for a genetic signal that shows up right across the modern populations of India, which means this is not a story confined to one isolated valley in Iran. It is a thread that runs much wider than that, because it turns out the Zagros farmers were genetically distinct from the other major farming population of the ancient Near East, a group known as the Anatolian farmers who lived in what is now Turkey and who were primarily responsible for carrying agriculture into Europe. These two farming populations, the Anatolian group and the Zagros or Iranian-related group, diverged from one another and developed along separate genetic paths for thousands of years before either one began expanding outward across the wider region. That distinction matters because it means there were effectively two separate ancient engines of expansion, both radiating outward from roughly the same broader part of the world, carrying overlapping but not identical genetic signatures with them as they went. And both of those expanding populations, it turns out, were carrying variants tied to the same intermediate olive-leaning pigmentation profile. When the Anatolian farmers began moving into Europe roughly 8,000 to 9,000 years ago, they brought the derived SLC24A5 variant along with them, and over the following centuries, they mixed extensively with the Mesolithic hunter-gatherer populations who were already living across the European continent. A detailed study of this mixing process turned up something genuinely striking. When researchers scanned the genomes of Neolithic-era Europeans looking for signs of strong natural selection, the single region of the genome showing the strongest excess of incoming farmer ancestry, stronger than almost any other region tested, was the area surrounding SLC24A5. The older hunter-gatherer version of that gene was being actively pushed out and replaced by the farmer version, and it was happening at a rate far higher than random chance alone could explain. In plain terms, this particular pigmentation genetics was not just riding along passively in the mix of incoming and existing populations. It was being actively favored, spreading through early European populations faster than almost anything else in their entire genome. But Europe was only ever the western half of this larger story. To the east, the Zagros-related Iranian farming populations were undergoing their own separate migrations, moving across the Iranian plateau and into the highlands of what is now Afghanistan, Pakistan, and the borderlands of Central Asia. This eastward movement connects directly to one of the most significant ancient DNA discoveries of the last decade, the genetic reconstruction of the population behind the Indus Valley Civilization, the Bronze Age culture responsible for building the great cities of Harappa and Mohenjo-Daro. When researchers sequenced ancient genomes connected to that civilization, including a landmark sample recovered from a site called Rakhigarhi in modern India, they found a population built from a mixture of two main ancestral sources. One was related to these ancient Iranian farming populations. The other represented a much older, separate South Asian hunter-gatherer lineage that had been in the region long before any of these migrations began. Across the broader range of individuals connected to the Indus Valley world, researchers found that the Iranian-related component ranged anywhere from roughly 45% up to as high as 80% of total ancestry, depending on the specific population being sampled, with the remainder coming from that older, indigenous South Asian lineage. There is an important nuance here worth pausing on, because it would be easy to flatten this into a much simpler and less accurate story than the evidence actually supports. The Iranian-related ancestry found in these Indus Valley populations was not simply a case of the original Zagros farmers picking up and relocating eastward as a single unified group. Genetic analysis shows that this South Asian-related lineage actually split off from the Western Iranian farming populations at a surprisingly early point, before agriculture had even been invented in either region. That means the population that eventually contributed to the Indus Valley world was itself a separate, long-diverged branch of the same broader Western Asian family tree, not a late-arriving colony of Zagros villagers packing up and moving east. Still, because both of these branches ultimately descended from the same deep highland ancestry, the same ancestry where these pigmentation variants had already taken hold, long before either branch existed as a distinct population, the intermediate, olive-leaning tone tied to that lineage rode along with both expansions. One branch moved west into Anatolia and eventually Europe. The other moved east across the Iranian plateau into Afghanistan and ultimately into the world of the Indus Valley. Same deep root, two very different directions. This matters for more than just the specific question of skin tone, because it pushes back against an older version of ancient history that treated the Iranian plateau as little more than a corridor. a place people passed through on their way somewhere more important rather than a source of major population movements in its own right. For much of the 20th century, the dominant narrative of the ancient world centered heavily on Mesopotamia, Egypt, and eventually Europe, with the Iranian highlands mentioned mostly as a backdrop rather than a main character. The genetic evidence gathered over the last 15 years complicates that picture considerably. The Zagros and the wider Iranian plateau were not simply a waiting room sitting between more important centers of history. They were an independent source of agriculture, an independent center of population growth, and an independent origin point for migrations that reshaped the genetic makeup of an enormous stretch of Eurasia, from the Balkans in the west to the Indus Valley in the east. The pigmentation variants at the center of this story are, in a very literal sense, one of the traceable fingerprints that migration left behind. The scale of that eastern legacy is genuinely hard to overstate. Researchers working on this population history estimate that somewhere between roughly 2/3 and 3/4 of the ancestry of all modern South Asians traces back to the same broad ancestral population connected to the Indus Valley world. That is part of why, when you look at the gradient of skin tones across the modern populations of Iran, Pakistan, Afghanistan, and northern India, you see something that roughly tracks the proportion of this ancient Iranian-related ancestry each population carries, fading and blending as you move further from the highland core and mixing more heavily with older, deeply rooted South Asian ancestry as you move further south and east. There is a real meaningful pause worth taking here, and here is where the mid-video reason to keep watching sits naturally, because if a single physical trait like this can carry a 10,000-year migration story folded into it, the same is almost certainly true of other traits and other quiet details buried in your own family history, waiting for someone to actually go looking. The picture is still not finished, because there's a third major population that carried these same highland genetics in an entirely different direction. The steppe pastoralists, the Bronze Age populations of the Pontic-Caspian, steppe closely associated with the early spread of Indo-European languages. These steppe groups are often talked about, in popular history at least, as though they were one single, uniform population. Genetically, they were not. They were a blend formed from a mix of Eastern European hunter-gatherers and a substantial genetic component from the Caucasus hunter-gatherers, that same highland population, related to but not identical with the Zagros farmers, who carried the early version of the SLC24A5 variant. When these steppe-descended groups expanded outward, moving west into Europe and east across Central Asia roughly 5,000 years ago, they carried fragments of this same highland genetic inheritance along with them, layering a second wave of related pigmentation ancestry on top of the wave that had already been spreading from the Anatolian farmers centuries earlier. This is part of why modern genetic maps tracking pigmentation-associated variants show such a broad, continuous footprint, stretching from the Iberian Peninsula in the west, across the Caucasus, down through parts of the Arabian Peninsula, and out toward the Tarim Basin in the east. It was never one single migration event. It was multiple overlapping migrations, all ultimately drawing from the same shared highland source population in Western Asia. Because of this long, largely unbroken presence in the region, the modern populations of the Caucasus, including Georgians, Armenians, and Azerbaijanis, along with a number of smaller ethnic groups scattered through the North Caucasus Mountains, carry unusually high levels of this ancestral Caucasus hunter-gatherer component even. Today, often more concentrated than in populations much further away that only received this ancestry through the diluted, several times removed inheritance of the later steppe migrations. This is part of why researchers studying the deep roots of this pigmentation lineage frequently turn to modern Caucasus populations, alongside the ancient genomes themselves, treating them as something close to a living reference point for what this highland ancestral genetic package looked like before it was carried outward and diluted across a much wider area. It is also worth pausing on the ancient farming population of the Levant, the region covering modern Lebanon, Syria, and Israel-Palestine, because it forms a third, genuinely separate lineage in this story, rather than a simple extension of either the Zagros or Caucasus populations. Genetic analysis has found that these early Levantine farmers do not share the same specific ancestral genetic markers as either the early Zagros farmers or the Caucasus hunter-gatherers. Despite the relatively short geographic distance separating these regions, Levantine populations, both ancient and modern, absorbed genetic input from multiple neighboring lineages over thousands of years, including contributions connected to Anatolia, to the Arabian Peninsula, and to later movements originating further east. That layered, complicated history is part of why the modern populations of Lebanon, Syria, and the wider Levant show such a wide internal range of skin tone, sometimes even within a single extended family, rather than one uniform inherited type passed down cleanly from a single ancestral source. Now think about what all of this actually means in human terms, beyond the science itself. A gene doesn't just sit quietly in a lab printout, it rides inside real people, on real journeys, through real hardship. Somewhere in a mountain valley in Iran, 10,000 years ago, a small population was adapting to a specific kind of light, a specific altitude, a specific balance of nutritional pressures, and that adaptation became part of the biological toolkit they carried with them into farming, into herding, into migration, into the founding of some of the earliest cities the world has ever known. Skin tone here isn't just decoration, it is functional. It reflects a genuine evolutionary compromise between two competing biological needs that every human population on Earth has had to balance in one way or another. Darker skin, rich in eumelanin, offers strong protection against the breakdown of folate, a nutrient essential for healthy cell division, particularly important during pregnancy and early development. Ultraviolet radiation can degrade folate in the bloodstream, and populations living under intense, near-constant sun exposure evolved higher levels of eumelanin partly as a shield against that risk. Lighter skin works in the opposite direction. It allows more ultraviolet light to penetrate the skin, which triggers the body's own production of vitamin D, something that becomes significantly harder to synthesize naturally as sun exposure drops, particularly at higher latitudes or during long winters. Populations living in the moderate, mixed sun latitudes found across the Caucasus, the Zagros, and the Iranian plateau were caught between both of these pressures simultaneously, neither living under the intense year-round sun of the equator nor the long, dim winters of far northern Europe. An intermediate olive-leaning pigmentation profile looks a lot like a genetic compromise between these two competing demands. A middle path that let a population meet both needs reasonably well without fully sacrificing either one. That compromise almost certainly helped stabilize this specific pigmentation combination in this specific highland region, tens of thousands of years before agriculture ever gave it a set of migration routes to travel along. This is also where a rare and genuinely important exception belongs, the kind of detail that keeps the story honest instead of letting it collapse into something tidier than the evidence actually supports. Olive-toned skin is not found only in populations descended from this particular Western Asian expansion. There are populations in parts of East Africa, in parts of Southeast Asia, and in parts of the Americas whose skin tone falls into a similar visual range, produced through entirely different combinations of pigmentation variants that develop completely independently in those populations. Biologists have a specific term for this pattern: convergent evolution, when separate populations, under separate selection pressures, operating on completely different timescales, arrive at a visually similar outcome without sharing the same ancestral genetic source at all. This distinction matters enormously, because it means skin tone by itself is never enough on its own to draw a genetic conclusion about where someone's ancestry actually comes from. Only detailed DNA analysis, looking specifically at the exact variants involved and the ancient patterns of genetic material surrounding them, can actually tell you whether two populations that both happen to have olive skin arrived there through the same ancient migration, or arrived there completely independently through entirely separate evolutionary paths that never had any historical contact with one another at all. This is one of the genuinely important lessons that ancient DNA research keeps reinforcing across nearly every physical trait it touches. A visible feature, a skin tone, a hair texture, an eye color, can look essentially identical from the outside, while resting on completely different genetic foundations underneath, built through entirely separate evolutionary histories. The only way to actually tell those two situations apart is to sequence the genome itself and examine the specific variants involved, rather than relying on how a population happens to look to the naked eye. Separate research into the broader genetics of pigmentation and tanning response adds one more layer worth mentioning here. A study out of a major research university in the United Kingdom identified roughly 20 genes specifically associated with how skin responds to ultraviolet exposure and tanning, and researchers found that only about half of those genes were already known pigmentation genes going into the study. The other half had never previously been linked to skin color at all. One gene in particular, called IRF4, had originally been studied almost entirely for its role in immune system signaling, with no connection to pigmentation on anyone's radar. It turned out to also strongly influence how skin tans and how much pigment gets deposited in response to sun exposure over time. This tanning response layer sits directly on top of a person's baseline pigmentation genetics, the kind carried by genes like SLC24A5 and SLC405A2 discussed earlier. That layering is part of why two people who share very similar underlying ancestry can still end up with noticeably different skin tones depending on exactly how their individual tanning response genes are configured and how much actual sun exposure they've experienced across their own lifetime. It is also part of why the same underlying ancestral genetics can look meaningfully different even within a single family. Two siblings can inherit an essentially identical baseline pigmentation profile from the exact same two parents, and still end up looking noticeably different from each other by adulthood, simply because one of them spent childhood summers outdoors constantly while the other stayed mostly indoors, or because a slightly different combination of tanning response variants shifted how efficiently each of their bodies responds to the same dose of ultraviolet light. Genetics sets the outer range a person's skin tone can occupy across a lifetime. Environment and cumulative sun exposure decide where inside that range a person actually ends up landing. What all of this adds up to is something bigger than a simple factoid about skin color, and it is worth taking a moment to really sit with what it means for how people think about their own family history. Someone with olive skin who grew up assuming their family's story was purely a Southern European one may actually be carrying a genetic thread that runs back through the Caucasus and the Zagros Mountains, all the way to hunter-gatherers living there during the final stretch of the last ice age. Someone else with olive skin who grew up in a South Asian or Middle Eastern household may be carrying that exact same ancient thread, only shaped instead by the very different eastward-facing migrations that eventually helped build the population base of the Indus Valley world. Neither person is more connected to that original ancient highland population than the other. Both are downstream of the same deep genetic history, reshaped over thousands of years by entirely different migrations, entirely different mixtures, and entirely different local histories, but rooted ultimately in the same original highland place. None of this is meant to erase or flatten the real cultural, linguistic, and historical differences that exist between an Armenian family, an Italian family, a Pakistani family, and a Lebanese family into one single, undifferentiated story. Those differences are completely real, and they matter deeply to the people who live inside them. No amount of shared ancient ancestry changes the fact that these are distinct peoples, with distinct languages, distinct histories, and distinct identities that deserve to be understood and respected on their own terms, not reduced down to a genetic footnote. What the DNA adds is simply a layer underneath all of that, a reminder that some of the physical traits people take completely for granted, right down to the exact tone of their own skin, can be traced with real scientific precision back through specific ancient populations, specific highland valleys, and specific moments in the deep human past, long before any of the modern identities built on top of that ancestry ever existed at all. It does not explain who these populations are today, and it was never supposed to. It only explains part of where one small, visible piece of their biology originally came from. It is important to say clearly and directly that none of this means skin tone can reliably predict anything about a person's character, their intelligence, their culture, or their destiny. Genetics can trace a pigment through 10,000 years of migration with real precision. It cannot tell you who a person is, what they value, or what they are capable of. Treating a trait like this as a stand-in for anything beyond its own biological function is exactly the kind of overreach that responsible genetic science actively pushes back against. And it is worth remembering every time a headline tries to turn a genetic finding into something it was never designed to prove. The research in this particular field is also moving quickly, and that speed is worth understanding rather than treating as a weakness. Ancient DNA research as a functioning scientific discipline barely existed 20 years ago. The extraction and sequencing techniques used to recover usable genetic material from bone that is 10,000 years old were still being actively developed within roughly the last decade and a half. And the sample sizes behind some of the specific findings described here, especially those connected to South Asia and Central Asia, remain smaller than researchers would ultimately like them to be. As more ancient remains are excavated, better. Preserved and sequenced using continually improving technology, some of the specific numbers described in this video will likely shift. Some population boundaries will probably get redrawn. Some of what currently looks like a relatively clean two-branch story will very likely turn out to have more branches once the remaining sampling gaps get filled in over the coming years. None of that is a reason to distrust the core finding here. It is simply how a genuinely young, rapidly maturing scientific field is supposed to behave as it grows. So, the next time you happen to catch a glimpse of your own skin in natural light, and you notice that warm, golden undertone that never quite seems to burn no matter how long you spend outside, remember that you are not just looking at a simple physical trait. You are looking at the tail end of a migration story that began in a mountain valley in Iran, or in the highlands of the Caucasus, roughly 10,000 years before anyone on Earth had figured out how to write a single word down. If a detail this ordinary, sitting right there on the surface of your own skin, can carry a hidden history this old and this far-reaching, it is worth wondering what other quiet threads like it are still sitting inside your own DNA, waiting for the right piece of research to finally uncover them. If that is the kind of hidden story you want to keep following, stick around, because there is a lot more of it waiting to be found, and we will be right here digging into the next thread of it with you.